So welcome everyone to this ICR2 session 2 on prostate cancer theranostics. We have a very interesting program. I just would like to remind those who are not so familiar with worms that we have a very nice website which was actually created by Josh, and it's just www.worms.org, and you can go there and get a lot of information. Okay, I will start the session just giving a short introduction, and then we will have presentations by Professor Hans-Jürgen Wester, by Professor Mike Satek, and by Dr. Aviral Singh. And we will allow 1 or 2 questions after each talk, okay? But we have to keep actually in time. I would like to start reminding you of the origins of worms, and that has a lot to do with Australia. And this is a slide from Ajit Paddy, which he showed in Levy in 2012, saying that the 6th World Congress in Sydney in October 1994 was actually the origin of worms, because the first International Radiopharmaceutical Therapy Colloquium took place there, and guess who was the organizer? Professor Harvey Turner, a little bit younger at that time. That was in 1998, the next opportunity in Berlin with Professor Biersack as president of the World Congress. And it was really stimulating to sit in a cafe or, you know, not lecturing, but just discussing things and exchanging things, and that was really very exciting. Also, Andrew Scott in Levy in 2012 gave a lecture, and he stressed the role of the WFNMB for linkage with worms and for facilitating liaison with worms on radionuclide therapy. I think we are still in the process of that. We will try to strengthen our relation and in the interest of patients and of concepts to go further. Now, theranostics. Short definitions is that cancers in future will be classified by molecular phenotypes. So you will not speak about prostate cancer, colon cancer, breast cancer, but this organ site will be a secondary classification, and the molecular phenotype can and will be determined by molecular imaging, mainly PET-CT, but probably also optical studies, using cancer type specific probes. And treatment will be targeted specifically against the tumor, which is what we call precision oncology. And I think that prostate cancer is a paradigm for this approach, as molecular radiotherapy is applied based on the molecular phenotype, namely PSMA expression of metastases as defined by PET-CT before starting treatment. Now, imaging agents were actually first developed by the group of Marty Pompa, and PSMA should more correctly be named glutamate carboxypeptidate 2. It also has folate hydrolase activity, but this is too complicated, so everybody is naming it PSMA, so it's not prostate cancer specific. You know that it is present even in breast cancer, in brain tumors, in many tumors with neoangiogenesis. We will hear a very interesting presentation by Professor Wester on a new concept. So we came from F18, which was first applied by Marty Pompa, then went to gallium-68, and now we are back to F18 and the twin tracers, which offers the opportunity to label the same molecule with gallium-68 or with F18. Now PRLT, PSMA radioligand therapy, started with iodine-131, and John Babbage and his group were the first to treat xenotransplant with iodine-131 PSMA molecules and showed stunning effects in animals. and later on the group from Heidelberg, Uwe Haberkorn's group, treated patients with iodine 131 PSMA and showed that there is a very good uptake and a very long half-life in tumor. That's exactly what you need for an optimal radiotherapeutic, that you have a high uptake and a long retention time in tumor, because then the area under the curves really gives you quite a high dose to metastases, which can be more than 300 Gray from one treatment in bone metastases if there is enough uptake. Now, our group in Bad Berka was really the first using lutetium-177 PSMA, and I reported the first results when we started in 2013. I reported the first result at the meeting of the Theranostics World Congress at Johns Hopkins. And that was, again, a very nice collaboration with Hans-Jürgen Wester's group, and we could show that there is striking effects even in patients with widespread metastases. You can get complete remissions. We did not have overall survival time, but median progression-free survival, which was very encouraging. And we published this paper in the Journal of Nuclear Medicine. And meanwhile, there are dozens of publications on PSMA radioligand therapy, starting with Weineisen from the group from Munich on the INT molecule, which can be used for imaging and therapy. So it's a really theranostic agent. In Bad Berka, we have performed now 762 cycles in 254 patients. And you can see that there is a high number of patients receiving 3 or more therapy cycles, actually up to 11. And we have seen stunning successes, like in this case, where actually the oncologist recommended androgen deprivation therapy and external beam radiotherapy to this lymph node metastases. So the patient received the treatment, and 1 year later, it looked like this. So the lymph nodes were effectively treated, but unfortunately, the cancer had spread and disseminated to everywhere. And then we treated the patient with 2 cycles of lutetium PSMA, and he had a complete remission with a PSA of 0.0. You all know the data from chemotherapy, hormonal therapy, and alpha-radin treatment, where survival benefit ranges actually from 2.5 to 4.8 months. So it's actually very limited, and especially with chemotherapy, we see also a lot of adverse effects. Our data now at 61 months starting treatment in 2013, we have a PFS of 9.8 months and actually a median overall survival, which is 30.9 months. Now, this is not a prospective randomized trial, right? This is a single center study, but I think the data clearly point to the fact that we can prolong life in those patients with very good quality. And if we look, when should we perform this kind of therapy? So we analyzed our data and showed that the median overall survival with previous chemotherapy is 38 months, and without chemotherapy is 19 months. So it just doubles if we move the therapy more in the earlier phase of the treatment. And this is again an impressive patient, an internal medicine specialist with a widespread metastases. He had a lot of pain and problems in walking actually, and had to give up his practice. So we treated him, and we can use the lutetium under therapy, also looking at therapy response. And again, he had a complete remission of the disease with a PSA of 0.0 nanogram per milliliter, and could restart his practice and even doing regular sports. And what is also remarkable is that we see this success continuing for more than 1 year in some patients. We will also hear about this very exciting ART or alpha radiation therapy from Mike Satek, and he was one of the first in the world also using bismuth-213, showing that there can be very nice responses in lymph node metastases. Like in this case, we also have treated a number of patients in Bad Berka, and to make a long story short, one can say the bismuth therapy is feasible. However, it is in the activities we use not very effective, and if you want to use higher activities or more frequent cycles, it becomes very, very expensive. So I think just by reasons of very high costs, bismuth is really out of the race, and actinium-225 must be considered the first choice agent, and that has been convincingly shown by the group from Heidelberg again. However, they found that xerostomia became the dose-limiting toxicity if treatment activity exceeded 100 kilobecquerel per kilogram per cycle, so roughly a dose of more than 10 megabecquerel. And we have looked at some ways to block the uptake to avoid this toxicity to the salivary glands. And we have published a first patient where we injected actually Botox. As you see in the upper row is before, and on the side we did not inject. And in the lower row there is a image of the site injected by Botox. Is not optimal because it has a too long effective time where it works, but we are looking at other agents now, and I'm sure we will find something. So the take-home message in a very short summary is that PRLT is really very effective in end-stage disease. It can improve clinical symptoms dramatically, and it prolongs progression-free survival, and probably also overall survival is really excellently tolerated by nearly all patients treated, so we do not see any or very minimal hematological toxicity in chemotherapy-naive patients. We do not see any nephrotoxicity. Okay, and I would say lutetium is not nephrotoxic. Even if you give 10 or 15 gigabecquerel, it is not nephrotoxic. We can discuss later why not, but we have seen no toxicity even after 5 years of treatment in these patients. Combination therapies are really needed. We discussed that already in the morning, and I think these radiohybrid tracers will be very, very interesting, especially for those centers having a high patient load, and new therapeutic radioisotopes, especially actinium-225, are very promising. Finally, we will need prospective clinical trials, and that's why I invited actually Michael Hoffman to present some data from Australia. Really, Australia is leading the way if it comes to clinical trials. And I'm very, very impressed how you got all these teams together, 10 centers, and you see that the recruitment is really fantastic. And you will tell us in more detail about these trials. And I think it's a model for other countries which we have to follow. This was taken in October 2012 when Melbourne was awarded the World Congress, and if you look carefully, all have become a little bit older, except Sejing Lee. She is looking the same as that time. And I will end with a statement of Fidel Castro, which he made in 1959. We all should retire relatively young. Thank you. Okay. So Hans-Jürgen, it's your turn, please.
Yeah, dear Richard, dear colleagues, I would like to present today the concept of radiohybrids, which is a concept that we have developed at TUM as a strategy to improve the current situation, and especially all the questions which arise during the last years with respect to F-18 labeling, how gallium tracers, especially, for example, PSMA tracers, will be developed in the future, whether they have a niche, and so on. Disclosure. When we are thinking about a new concept of F18 labeling and how this fits to the current situation of what a lot of people say theranostics means, the combination of gallium and lutetium for labeling of one molecule, then we should go briefly into the past and should take a look why this close link between gallium and lutetium exists. It's not that this is inherent, it's a chemistry issue, a chemistry limitation that occurs very early in the development of the somatostatin traces, where first indium traces have been used by Krenning and Basel group, by Helmut Mecke, Jean-Claude Reuby, Lancet publication, starting with the iodinated probe, then very early the lutetium compounds. And then in parallel, a lot of people thought about PET imaging. And Peter Smith-Jones and Helmut Mecke presented these DFO octreotide in 1993. And in Jülich, that was in fact my PhD thesis, we already developed F18 octreotide. But at that time point, that was a very complicated synthesis, and nobody would like to do this in a way that this, because this needed 3 steps, 120 minutes. So finally, we published these data. The tracer itself would had a high uptake in the liver, so lipophilicity was not favorable, but independently, compared to that which was at that time point already available with gallium, which was also not compared to that what was later available, this paper concluded that the gallium traces are better with respect to the labeling strategy. And the same in the same year, publication from Helmut Mecke and Peter Smith-Jones concluded exactly in the same way. Okay, we have a gallium complexation. F18 is complicated. The production is complicated. So this favors gallium. That finally installs the strong link between gallium and lutetium. Also, we are coming from F18 as the most often used PET tracer. And F18 FDG is the cheapest tracer we have. One dose maybe is in the range when high doses are produced in the range of 27 to maybe 35 euro. So finally, this resulted in 20 years later, in tracers that now are approved in Europe and the US. And when we started now with the PSMA, we did the same issue, we did the same strategy, going with the gallium tracers first, then doing here these therapeutic agents. And of course, then the discussions with F18 tracers again came up, but we used the same strategies, business as usual. We did not take care about what happened in the last 20 years and whether we can use these improvements to making it even or much better. That is exactly what we try to do. So we have now all these traces, and all of these traces have their advantages and are good or are excellent. But that's not, maybe that is the current situation, but can we do it in a better way? And we have current questions which we should address. Is gallium radiopharmacy a long-term alternative to F18 radiopharmacy? Is there a niche for a variety of future gallium traces or F18 traces in the field of PSMA, or for example also somatostatin, folate, and whatever exists? And is it possible to use PET for therapeutic dosimetry, and how we can do this? When we're thinking about this, we should hold a while and should think, what is that what we really would like to have? What would we like to have in an ideal world? And that is really a question you should try to address. And when you go through this, through all these tracer characteristics with respect to cost effectivity, whether some would say we need F18, of course, the others would say we need the gallium tracers, we don't want to completely switch to F18. Very simple production, synthesis time, at least like FDG, yields high, should be transferable, should be adaptable to other traces like somatostatin, folates, as mentioned before, excellent biodistribution, imaging time after 1 hour. So how we can reach this? How do others solve such a problem? Let's take a look at a different technique. Hybrids. Hybrids, an established technique coming from, let's say, going from an established technology to a new technology without skipping the best of the old world, combining the best of the old world with the best of the new world. To have, for example, in a luxury car, two strategies how to run this car. From outside you cannot distinguish, but these things are inherent in this frame. And exactly that is what we did with the PSMA. And this is transferable also to other peptides because PSMA inhibitors are peptide-like traces. So what we did, we combined in this PSMA inhibitor-like molecule inherently a labeling site for radiometals and a labeling site for fluorine. So that in all these, or that for all different applications, these strategies are available. What we now have are really kind of monozygotic chemical twins. When we have, for example, the cold F19, then we have a hot radio metal, or we have the opposite, a hot F18 and a cold radio metal. But these traces, since they are now really identical monozygotic chemical twins, exhibit exactly, absolutely identical affinities, selectivities, lipophilicities, and pharmacokinetics. So that means that, for example, when our group, in the meantime, we have worked with one of these traces and have made several hundreds of patient scans with the fluorine compound, that these data can directly transfer to those groups who would, for example, use the same compound for gallium-68 labeling while using the cold F18 labeling precursor. And the same can be done, for example, when combining F18 with a therapeutic isotope, here in a way that either this can be used, this tracer then can be used for pre-therapeutic imaging, and this then subsequently for the therapy. So when we're focusing now on the F18 option, why is this a breakthrough? It's a very complicated, maybe very complicated synthesis for the F18 part. No, it is not. We really took or implemented the best currently available technology. That means that we have very short labeling, very short synthesis times. We can do the labeling at room temperature for approximately 5 minutes. So we have no decomposition, no byproduct formation. We can use simply solid phase extraction purification like a gallium labeling methodology. This cassette looks very similar, high specific activity. We are producing these compounds in yields or in overall activities of 50 to 100 gigabecquerel. This is a cassette, very simple, as mentioned before. F18 labeling at room temperature for 5 minutes. There is no azeotropic drying. We do this on a cartridge in a very simple manner, IP protected. Overall synthesis time is 800 seconds, ready for application. And the yields are 60% in this automated manner. When we do this in hand, this is not finally optimized. It's already optimized, but there is still space for improvement. If we do this manually, we reach even 90%. When we're checking and when we're comparing these new compounds, and we're calling them radiohybrid PSMAs, and these are different versions, and the dark blue are the gallium-68 cold F19 radiohybrid versions, and the light blue is one example of a lutetium-177 cold fluorine radiohybrid molecule, compared here with the DCFP-YL, or with the currently used DKFZ 1007. You see that with respect to the lipophilicity, these compounds are somewhere between these two. So that means that we do not expect full uptake in the liver, or not excretion like we have it in DCFP-YL, but something in between. That are the affinities of these compounds, again, compared to our internal standard. That is the iodobenzoate of the KUE inhibitor, DCFP-YL, and DKFZ 1007 here. So the IC15 are even better than those, or in the range between 1007 and DCFP-YL. So we can expect that we have very high uptake in tumors, similar to those traces which are already established and used. With respect to the internalization, we are similar or even better for some of these traces compared to these two here investigated in the same assay for enzyme-mediated internalization into the cells. That is the biodistribution in mice. And again, we see here the three compounds which we compared, more or less same uptake in different organs, somewhat a lower uptake in kidneys, with respect to tumor uptake, somewhat higher, but this is more or less, at least in animals, within the error range. When, however, we take a zoom, then we see some differences, and that is exactly what I mentioned before with respect to the lipophilicity. Here you see the biodistribution in man for the 1007 compound, and here for the DCFPYL. And we are somewhere in between the lipophilicity. That means that we do not have this strong liver uptake we have for the 1007, and we do not have this strong bladder activity excretion as we have it for the DCFPYL. We are somewhere in between. That means lower liver uptake, but hopefully no uptake or no excretion into the bladder, but kidney accumulation. So in summary, before we go to the first patient examples, we have really more or less said what we expected, what the ideal word would represent. High affinity, optimum log P, high internalization, no azeotropic drying, fast synthesis, labeling at room temperature, 5 minutes, 60% yield, very simple production, low hardware components and requirements, high specific activity, really high specific activity. And that is now, or that are the first examples. I cannot show more than those due to IP protection, but you see here, and these are really representative to several hundreds we have done in the meantime in Munich, together with Matthias Eiber and Markus Schreiber and Wolfgang Weber. Here a patient, and you clearly see the lesion. This is a primary prostate cancer, so here no uptake in the bladder, medium uptake in the liver, as mentioned before. And here an example, PSA of 0.9, clear delineation here of the metastasis or the lymph nodes, and very low to negligible uptake in the bladder in this patient, and more or less the same here, PSA 1.1, a picture in this patient. So exactly that what we expected happens, that we have a tracer with a biodistribution in between 1007 and DCFPYL can be labeled in a very effective manner. And when we now take a look, that is what we obtain now with the F18. But now, again, we can use the same compound to be now introduced, for example, in the synthesizers you are using for gallium labeling, and then you have exactly these traces, but as a gallium tracer with a cold F18. That means that based on this F18 tracer, we can simply substitute the isotope and use the free chelate for gallium labeling, and then would obtain exactly the same biodistribution because it is the same compound. And then we would get rid of this bladder activity as we obtained in the currently used gallium tracers, and you would have this kind of image, what we obtained with the F18 compound. And as I mentioned before, of course, this system can be translated to other peptides, and that is what we are currently doing. We are adapting this to somatostatin, to 6CR4, to GRP, to folate receptors, and to other peptide and peptide-like tracers. And of course, that is something I would like to investigate and would like to see whether this is really something that works, that we can add a PET option to the radioligand therapy in a way that we have now a tracer with 120 minutes half-life or 110 minutes half-life that allow to make PET imaging up to 6 hours post-injection, maybe to 8 hours post-injection, and then to have a valuable estimate or measure for the dosimetry of the corresponding lutetium agent. And as mentioned before, and that's going back to the pictures I showed before, these dark or light blue bars are those for the lutetium, hot lutetium cold fluorine compound. And you see that even higher, the hydrophilicity is somewhat higher, so the bladder excretion should be higher. That means really excretion, fast excretion, which should be favorable for the dosimetry. The IC50 is in the same high range, and the internalization is the best of all compounds. So that are really very good characteristics for the first cold F19 lutetium-177 compound for therapy, which can be used as PET agent for pre-therapeutic dosimetry, as already investigated for gallium, whether this is possible or not, in publications that have been recently been published. So overall, what have we done? What we have? The current situation is that we have radioligand therapy and gallium radioligand imaging in a way that we are using the same compound to be labeled with two different isotopes. F18 compounds are completely separated, are different synthesis, more complicated, and that is where we say this is our theranostic concept. What we now have done, we have inherently added an F18 option to both of these in one molecule. So really combining therapy with PET imaging. And that in a way that we can simply switch to whatever kind of option we would like to have. And that means that we're really linking the therapeutic and the diagnostic option with the cheapest PET isotope which is available. And so in sum and in conclusion, I think that radiohybrid traces are not a compromise. I think this is very important. It is not a compromise that we add something which inherently result in a compromise where we have to handle some negative effects. No, the sum is really more than the single tracer option can offer, and therefore it is a really synergistic approach. And overall, as you see here, radiohybrids can offer a variety of different new strategies with respect to theranostics. Thank you for your attention.
We will proceed to the next paper, which will be given by Professor Michael Hoffman on meeting the challenges of randomized multi-center clinical trials.
Thanks, Richard, for the invitation to talk to you today. So this talk's going to be a little bit different. It's going to be a bit of a focus on how we have undertaken some multi-centre trials in Australia that are currently underway and the steps in order to do that. Richard and I sort of both grew up in a similar way with lots of retrospective studies, and I'm a little bit younger, and what I learnt is it's very hard to get these published in anything but nuclear medicine journals. We would have some really striking data showing amazing responses in both neuroendocrine tumours or prostate tumours, but I would submit a manuscript to an oncology journal and they would not even send it out for peer review. The oncology journal would say this is single centre, retrospective data. It looks very good, but if you really believe it, resubmit it when you've done it prospectively. And then we've got the alternative, which is the NETA-1 approach, an industry approach, where they've done a modest-sized trial of 200 patients or so, and it's got into the New England Journal, and that's perhaps what we should be aiming for. And there's a hybrid in between that NETA-1 industry model, where a trial costs $30 to $50 million to perform, to the retrospective trial, which is just done using clinical care, and that's what the talk today is going to be about. So we've currently got two large trials up and running in Australia, and one is an imaging trial, which is the one I'm going to focus on to start with, and then the second trial is a therapy trial. So this is the Pro-PSMA trial, and it's now activated at 10 centres around Australia, and it's a prospective randomised trial of the impact of gallium-68 PET-CT imaging for staging high-risk prostate cancer prior to curative intent surgery or radiotherapy. And you can see the sites up and running around Australia. And there's a lot of investigators involved from nuclear medicine, but also surgical oncology and radiation oncology. And most sites have an investigator from all three disciplines. So this is the trial design. We take patients with untreated biopsy-proven prostate cancer that are being considered for curative intent prostatectomy or radiotherapy. High risk is defined by PSA over 20, Gleason grade group 3 to 5. This is the new Gleason grade group system, or clinical T-stage over T3. And then they're randomised 1-to-1 to either have a PSMA PET or conventional imaging, which is a CT and a bone scan, in order to get patients to participate in the trial and really to optimise their management. There is a crossover component. So if you have a CT and a bone scan and it shows less than 3 distant metastases, then you cross over to the other arm, or vice versa, so that you're not missing out. The patients are managed, and at 6 months we do follow-up, including repeat imaging, and at this time point we determine whether the baseline findings were true or false, and to get some other secondary endpoints, we continue to follow up patients with negative scans to see how good was a negative conventional imaging or PSMA PET. So the primary endpoint is accuracy, and the secondary endpoints are management impact, health economics, radiation exposure between the two arms, reporter agreement, and safety. And this is a grant that was awarded back in 2016. We randomised the first patient about a year ago, and you can see the recruitment is going very well. So as of a few days ago, we had randomised 150 patients out of 200, and all 10 sites are activated. And this is the recruitment by site. So the first few months, we just had one site involved. And over time, in the last few months, lots of sites involved, all contributing patients. And we're actually going to expand this to a 300-patient trial because it's running so well. So the question is, how did we get there? How did we get to this point where we can run a big multi-centre trial using an academic model? This is not an industry trial. And the rest of my talk is really going to run through how we've got to this point so that hopefully other people can replicate what we've done. So I think clinical trials are launched from pilot studies. So all the retrospective data we do is very valuable because no one's going to embark on a multi-centre trial of PSMA PET if there's no images available. We need those early images from Heidelberg showing us that this is an extremely good tracer. We do need some initial data to get people excited. Otherwise, no funder is going to say, let's spend a good amount of money on a multi-centre trial. In order to really get grants needed to do a multi-centre trial, we need some of these things. We need some data. We need expertise in writing protocols. We need to be able to prove that what we're going to be doing in the trial is feasible. So we need some of the assumptions are tested. We need to know that the costs are reasonably accurate. We need to develop collaborations amongst the centres so that we can work together. No one will give us money without a track record, so it's important to have a good number of publications under your belt and have published these so that people have confidence that you're able to do the research that you say you can do. It's really a team sport, and I've divided into these 4 components. Obviously, the investigators. We need nuclear medicine people, not just physicians, but also chemists, pharmacists, technologists, physicists involved. But we need to involve our clinical specialists, which in this case is surgeons for this trial, since they're the primary referrers. And it was a really important component to get our urologists on board with this study as part of the grant, and that involved getting urological societies to perhaps get involved in the protocol writing. We need organisations, because individuals alone, when you apply for a very big grant, it's a little bit disempowering for the person that's going to give you the money to give it to 1 or 2 people. So it is useful to have a reliable institution, but also clinical trials groups, networks that have experience in rolling out these clinical trials to make them happen, and industry partnerships as well, since the products we're using are often come from industry, and we need to get some of the API or some of the products sometimes for free in order to do our trials. We need a coordination centre that has a study coordinator, a good statistician, and nowadays we need to do trials according to GCP, which is good clinical practice. So you need to have perhaps done a course in GCP and know what that involves. We need databases created, and this needs to be independent. You don't want the chief investigator collecting all the data himself, because that's subject to bias. So we do need someone independent doing this. Obviously, they need a grant in Australia where there's a number of sources. NH and MRC is our big public source of grants, but increasingly there's philanthropy, industry, or institutions that are willing to fund this. And we need a principal investigator. We can have all these elements, but it really does need one person to take on this project and drive it and get everyone together. Without the sort of PI driving it, I think it's hard to put this all together. So one of the things that's made this trial work was ARTNet, which is the Australian Radiopharmaceutical Trials Network. This is a collaborative network incorporating medical specialists, techs, scientists, researchers with a shared interest in multidisciplinary clinical trials that use radiopharmaceuticals for imaging or therapy. And their mission statement is to promote and facilitate innovative, collaborative clinical research utilising radiopharmaceuticals. And they're part of a broader Australian Clinical Trials Alliance. So this consists of all the groups such as the GI oncology, brain oncology. I think there's sort of 30 or 40 different clinical trial groups under this umbrella organisation. And when you're applying for a grant, if you've got a clinical trials group on board, I think that gives your proposal a lot of authority, but also reassurance that it's going to happen and it's going to be feasible. So this was the application process for the Pro-PSMA trial. I just thought I'd take you through it. It's going to be different for every trial depending on the pathway. But I just thought I'd give you a good overview of how we got to that point. So there was a grant from the Prostate Cancer Foundation of Australia and Movember. Movember's a charity, and PCFA are not that good at necessarily getting the money together, so the money came with Movember, but PCFA are good at evaluating grants and doing that. So there was a partnership between these two organisations for a $1.5 million clinical trials grant. And in order to sort of get your foot in the door, you just needed to do a 2-page summary, which was fairly easy. A scientific abstract, a lay summary, and some coordination support details. It doesn't take too long to put this together, but having that art network was valuable at that time point, just to get everyone together quickly and put this proposal together. After that was submitted, we were shortlisted and then asked to provide a detailed proposal. So this was more like a conventional grant, an 18-page protocol, details of which sites will be involved, co-investigators, CVs for all the co-investigators. At that time point, we engaged collaborators in more detail, which was some health economist, a consumer advocate, which is increasingly important to have involved in these trials, to have lay people involved, ArtNet, and some other groups, oncology clinical trials group, a radiation oncology clinical trials group, and our surgical oncology clinical trials group. We needed someone to actually take the money, which was University of Melbourne, and we needed to put together a detailed budget. We were then shortlisted as one of, I think, the 2 or 3 preferred projects, and there was then an interview stage, and Movember and PCFA actually got 3 international experts from overseas to fly into Australia to give us a bit of a grilling and take us through our grant, and we had to do a PowerPoint presentation and then answer any questions. So it was really quite rigorous. And the panel was a medical oncologist, a radiation oncologist, and a urologist. And we were represented actually by 3 people, myself, the key urologist driving the trial, which was Nathan Larenchuk, and the third person was actually a patient advocate who had had a PSMA PET scan and was really passionate about this technology and gave his consumer perspective on why this trial was important. And after all of that, we were awarded the grant, and we were asked to revise a few things and give a revised protocol, and then the work really began. So a lot of work to get to that point, and the funding was awarded April 2016, and you'll see it took just under a year to get from that point of there to developing the detailed protocol to randomising the first patient. And these were all the things that needed to be done to get the trial up and running, and I won't go through that in too much detail. So just to focus on some nuclear medicine aspects of this trial, prior to commencement, all the trials have to image a phantom with gallium-68, and we also got together with ArtNet, came up with some gallium PSMA production criteria, because every hospital does things a little bit different. These are generally all made on site in the hospital radiopharmacy, so we came up with some standardised quality assurance. And during the study, we have all the PET scans second read by a core lab, and we also have some off-site data monitoring by the group running the clinical trial. In terms of this is an unapproved product, gallium PSMA 11, and this is something that varies by country, and we're quite lucky in Australia because we have what's called a Clinical Trials Notification Scheme, which is the CTN scheme from our Therapeutic Goods Administration, TGA, which is like the US FDA, but this is a notification process. In the US, if we were doing this trial, I understand we would need to sort of give the protocol to the FDA. They would go through it. It would involve a number of discussions back and forwards. In Australia, it's just a notification process. We need to tell the TGA that we're going to use an unapproved therapeutic good, but they essentially rely on the Human Research Ethics Committee to do the review of scientific validity. The TGA don't actually evaluate the clinical trial at all. And actually the institution is the approving authority that gives the final authority to conduct the trial. So this gives us a little bit more flexibility. We don't need to waste time engaging the TGA to review our protocol We just go through the usual protocol of ethics, and once it's approved, we can just tell the TGA we're doing this trial, and they note that and record it. So they're trusting us to some degree. We came up with some standardised QC for gallium PSMA production, and this involved HPLC, which many centres were not using at the time as a routine production method, and I won't go through this in any detail, but we said you have to have 3 consecutive successful runs with full quality control in order to be eligible to participate in the trial. And then for routine batch production, the QC is a little bit less, but still involved HPLC. We also did some PET scanner validation. This involved a nemaphantin imaged with gallium-68. I've got to be honest, I thought this was a little bit of a overkill and a waste of time because many centres had already done this for fluorine-18, and I thought, why bother doing this for gallium-68? It's a little bit of a waste of time. It adds a bit of cost to the study. But being a multi-centre randomised trial, we did feel that robust QC was required. So we did this, and much to our surprise, almost every centre failed the QC, including our own. And what we found is that 10 out of the 14 PET systems actually underestimated the SUV by 15% on average. 15% was outside of our sort of 5% acceptance window. 15% is quite a lot, and the reason for this needed to be investigated. And we actually identified it wasn't the PET cameras that was wrong, it was actually the dose calibrators. So most centres around Australia were using the same dose calibrator, and although they were measuring fluorine-18 correctly, they were actually giving erroneous measurements for gallium-68 by 15%. So we needed to adjust all the dose calibrators so that they were measuring accurately. And this was actually a dose calibrator manufacturer sort of error in the book that they were shipping to the sites. So this highlighted the importance of testing the entire chain of measurements and calibration as part of sort of a pre-qualifying site assessment. So this unexpected finding has been written up, I think in this month's or last month's J&M. So you can pull out this journal article, but it's one of the first publication that has come out of this trial. All the PSMA PETs in this trial undergo central review. So the DICOM data is de-identified, sent to a core imaging lab. If the local read does not equal the central review, we then discuss the results. We had to, what about if the central review and the central reader can't agree? We decided that if a consensus cannot be reached, the patient will be managed according to the results of the local reader. That's because they're most in touch with their referrers. It's hard for a local nuclear medicine person to sort of tell their urologist, the central site disagrees with me, you should go with what they say. So we decided that the local reader will be the sort of arbiter in the end. But this allows us to look at that secondary endpoint of reporter agreement in a very robust way. So that was really the first part of my talk on the pro-PSMA study. And as we were getting that study underway, another grant opportunity came up, which was also through the Prostate Cancer Foundation of Australia, but instead of in partnership with Movember, it was in partnership with ANZUP, which is the Australian and New Zealand Urology Clinical Trials Group. And they also came up with a $1.5 million grant to look at sort of some higher risk strategies for treating patients with metastatic prostate cancer. So we submitted a proposal to do a randomised trial of lutetium PSMA. And we'd already done a lot of the hard work in the pro-PSMA trial of developing that network, standardising the gallium PSMA production across 10 sites, which we thought we could transfer across to lutetium production. And we were awarded the funding for this trial, and I'm not going to go through the same level of detail about how we did that, but this is the TheraP trial, which is short for actually Theranostics PSMA, and it's a randomised phase 2 trial of lutetium-617 Theranostics versus carbazitaxel chemotherapy in patients with progressive metastatic castration-resistant prostate cancer. And again, the partners here are PCFA and ANZUP, but also ANSTO, who make the lutetium-177 and actually provide the lutetium for free as part of this trial to study sites, and now Endocyte, who make the PSMA-617. It was ABX prior. We have a big clinical trials research centre involved in this trial that are involved in doing really very large, high-quality trials. We've got ArtNet involved, and there's also money from Movember which has come to do this trial. So this is the study design. In brief, we take patients with metastatic disease who are potentially suitable for carbazitaxel. So these are patients where the oncologist is saying you are eligible for carbazitaxel. They must have a rising PSA. They have to have had docetaxel in the past, ECOG 0 to 2, adequate renal, bone marrow, liver function. They have to have a PSMA and an FDG scan showing that the patient is suitable. And then they are randomised 1-to-1, either to carbazitaxel or lutetium 617. This is a 200-patient study, open label, randomised. There are some stratification factors, 1 by study site, 2 by volume of disease, less than or greater than 20 sites, and whether or not you had prior abiraterone or enzalutamide. So that stratification means that we'll have sort of an even balance of these 3 items amongst the 200 in either arm. The lutetium is given as 6 cycles every 6 weeks. We went with a de-escalating dose schedule. So the first dose is 8.5 gigabecquerel, then 8, 7.5, 7. If you get to cycle 6, you'll be given 6 gigabecquerel for cycle 6. And an important element of this trial is that we've incorporated a sort of intermittent strategy. So if you have an exceptional response, your PSA drops massively, your PSMA signal goes away, we will stop treatment with lutetium, but you don't go off trial. You stay on the trial, and when your PSA starts progressing, you can then continue up to the 6 cycles. So this is quite a unique concept to put in a randomised controlled trial, and we think this is a key component to make this arm optimised. Again, being a multi-centre trial, we have some central QC done by ArtNet. We've just copied across the gallium PSMA QC from the Pro-PSMA trial. We had to develop some new QC for the lutetium component, and we have the PET camera validation also from the Pro-PSMA trial. These are our lutetium QC specifications, which were really developed in collaboration with ArtNet, their team of radiochemists and pharmacists at the time, so it's fairly standardised. This is a brief version, and we also have some minimum production methods since each site. We'll use some different methods. Some sites are manufacturing this manually, some sites are using Syntomics or Eker and Ziegler automated synthesis units, but everyone needs to use the same ingredients and everyone needs to meet the same minimum QC. All patients have both a PSMA and an FDG PET scan to determine eligibility, and these all go to central review. For this study, we use an online system called Widen, which was developed for some of the lymphoma trials, to do our sort of central review. If there's disagreement, we sort that out with consensus discussion like in the Pro-PSMA study. So this trial has begun. It's now activated at 4 centres around Australia, but it will be activated in 10 sites around Australia. Many of these sites are the same sites as involved in the Pro-PSMA study, but there are some differences. This is the progress to date. It's really just started. We've registered 14 patients and we've already randomised 8 patients, and it looks like we are going to schedule. So I'll leave it there, and if there's time to take any questions, I'm happy to do so.
Thank you very much, Michael. I think you have shown that if you do once the exercise, the second time is easier.
Correct.
Really have to congratulate your team effort, as you said, that you have succeeded in such really prospective trials. You have a little bit run over time, so one question, urgent question.
This 200 is a magic number, or you have done some outcome analysis like 80% power, 85%, 90%, what is the basis? Because 200 is a magic number.
Oh yes, 200 is the magic number in both studies. And no, we have done, there were biostatisticians involved and a lot of discussions around power calculation and study. So I don't have those numbers off the top of my head, but there were some serious power calculations done. But in reality, you are restricted by funding, so you need to balance that and endpoint. So you'll note in the TheraP trial, the primary endpoint is not overall survival, but it was PSA response. I wanted it to be overall survival, me personally, but the biostatisticians did not feel that it was necessarily powered for that. I actually confident that we'll still show
Yeah,
a survival
definitely.
benefit with 200 patients, but the statisticians
We're very careful.
are very careful.
Okay.
Is there any stopping rule? Is there any stopping rule or interim analysis?
There is no stopping rule. Actually, for the Pro-PSMA trial, we've actually expanded it from a 200 to a 300 patient study because we have done some interim look at the positivity rate of the PSMA PETs, and there's a few more negative studies than we expected, and we thought the study was a little bit underpowered. So that's going to be expanded to a 300 patient study very likely, but there's no interim stopping rules.
Okay, thank you very much. We have to, I'm sorry, we can maybe discuss in the break, but we have to continue. We come to the last paper of this session, which is given by my co-worker, Dr. Aviral Singh, on new radioisotopes for theranostics of prostate cancer. Aviral, please.
Good day, colleagues. Thank you, Chairperson and the organizing committee for this opportunity to speak today. So, as we all have seen, that the treatment of localized and metastasized disease, primary treatments are surgery and external radiotherapy. We've also seen what nuclear physicians are doing in the diagnosis and eventually following this up with targeted radionuclide therapy. So we know that prostate-specific membrane antigen is essentially membrane-bound type 2 glycoprotein, which Professor Baum had introduced earlier in the talk and spoken about it. But what's important is that it is overexpressed in malignant prostatic tissue, and the higher the dedifferentiation and the more complex the disease gets, the higher the expression of PSMA becomes. We've already seen how actinium, one of the newer radionuclides on the block, acts and works for us in this in theranostics of prostate cancer. However, from a radiopharmaceutical perspective, although I'm not a radiopharmacist, but it could be that with the short half-life, whether the recoiling daughter radionuclide detaches from the target agent as a consequence of alpha decay and maybe not actually being effective in certain cases. Therefore, newer and better chelators need to be developed for more stable coordination chemistry of actinium with PSMA. Now, talking about futuristic or certain matched pairs of radionuclides for theranostics of PSMA, there's a whole list of publications that can be seen, and these are primarily our collaborators from PSI Willigen. So here we see radionuclides which have alpha therapy, then emit Auger electrons, then beta therapeutic, then for positron emitting, and also for SPECT. So mainly, let's first stop at scandium, and with regards to the logistics of it. Now, gallium, as we know, has a 68-hour minute half-life, whereas compared to that, scandium has a longer half-life of about 4 hours. Now, what this could translate into, and especially for people where they do not have radiopharmacy facilities in smaller areas, where they want to pursue theranostics, but there is no generator available or it's too costly. It is possible to actually distribute scandium over very long distances. We ourselves received our scandium for over 6,000 kilometers transport by road. Now, this is one of the comparison of the image resolution comparison. A phantom study compared gallium-68 to scandium, and the resolution itself looks a bit better compared to the gallium. For production, there are two routes for the production of scandium-44. The first, a generator produced, where Professor Baum performed the first in-human study as well at our center, and the gallium-produced scandium-44 was provided by Professor Roche. However, the issues with generator production, so the positive things about generator production is that you can have it on site. You don't need a cyclotron. However, the production is extremely costly, and because of the extremely long half-life of titanium, it can be an issue with regards to radiation protection. Now, talking about cyclotron-produced scandium, you get a fast production, high yields, and a very high purity, but again, you need to have a cyclotron. Now, in short, cyclotron-produced, so scandium in the cyclotron is produced by bombarding protons on a calcium-44 target to achieve scandium. The separation of scandium from target material can also be performed, and it takes about 20 minutes time. With regards to imaging and therapy, so here you'd be replacing the gallium-68 with the scandium, and then using this as the diagnostic part of theranostics, and then coupling it with lutetium PSMA therapy. If you compare the cell internalization in preclinical studies, and that would compare the prostate cancer cell lines which are PSMA positive to the ones that are PSMA negative, you can see that the uptake internalization here at early 2 hours and 4 hours in the positive cells is much higher and almost similar to most of the other radionuclides, whereas compared to the ones where there's no PSMA expression and there's no internalization or uptake. So essentially, with regards to scandium, it has a comparable uptake and internalization of all PSMA radioligands, including scandium and lutetium. It is the, and again, as one can see, the specific uptake can be demonstrated by using positive and negative cell lines. With regards to biodistribution studies, again, if you see the, over time, if you see the uptake in the, PSMA positive cells, with time you can see an equal uptake, and similar, on the contrary, in the negative cell lines for PSMA, there is not, there's a very minimal uptake in the initial early time period, but then there's almost negligible uptake over time. With renal uptake as well, initially with the scandium, because essentially we have seen in clinical studies as well that there is higher uptake of scandium in the kidneys, even on the later time points. However, when you see with regards to pairing scandium with lutetium, one can see that the uptake in the kidneys over time reduces, and it is almost equal to the lutetium. So one can also think about predictive dosimetry when you're planning therapy. For biodistribution in all ligands here, you can, I'll just point out that in comparison to, say, for example, gallium-68 PSMA-11, which has a relatively higher kidney uptake and then washout, the scandium is more or less similar to the other ligands. Then these are some preclinical images obtained comparing PET-CT and SPECT-CT. So with PET with scandium, SPECT-CT with lutetium, and then compared to comparator gallium PSMA ligands. Again, preclinical studies over time, where you can see the, so this side here in the mouse is the one which has the PSMA negative cell line tumor in the mouse, whereas these are the ones which show PSMA expression. So over time, even up to 4 hours, you can see that there is a significant tracer that can be visualized. The first clinical study of the first clinical translation and first human use has been already published by the Bond Group, if somebody is interested. This is at the center, this is one of the first in human images using scandium PSMA 617. And it's pretty comparable in uptake to the gallium-68 PSMA. If anything, certain lesions could be better visualized, and if lesion per lesion count, there are more lesions visualized. And then these are the lutetium post-therapy scans. And if you see the distribution, say, for example, in this lesion, this is more or less similar compared to the gallium. So to summarize, so the potential uses for scandium would be that since scandium shows equal characteristics to lutetium PSMA in vitro and in vivo, and this could then translate into your pre-therapeutic dosimetry or treatment planning. Scandium also has excellent characteristics for PET imaging, and it allows for delayed imaging. So if you are suspicious about certain lesions or clinically there's something relevant, if on the early images you're not able to visualize, it is easier for you to perform with scandium later images or even on the next day sometimes. It is very easy to transport this, so this is certainly an advantage in areas where you do not have cyclotrons or you do not have a radiopharmacy to harbor generator. So it is possible that scandium could be used in tandem in future with lutetium or also as a theranostic pair with scandium-47. Good, I'll skip that one. But just saying that scandium-47 actually, it's scarce for now, but it could also be produced in a reactor. However, the concerns are the emission of high energy gamma rays, which may be actually a disadvantage to the personnel working and also the patient. So this is again imaging to show that in cases where there are, so I'll just go back to the slide, the issue with the emission of high gamma rays of high intensity, this could be bypassed with the use of the possible use of scandium-43 instead of scandium-44. And on these studies, you can see again, so this is scandium-43 PSMA, this is scandium-44 PSMA, and again here, scandium-47. So these are for imaging here, whereby using scandium-43, you could avoid the high gamma energy radiation exposure, and as a theranostic spare, scandium-43 could be used with scandium-47. Now for the next pair, this is a more wider range of applications with terbium, which has here 4 isotopes that one can see. So 3 of the first ones that you see have been produced in collaboration with Isolde CERN in Geneva, whereas the terbium-161 is produced at PSI Willigen, where our colleagues are working. Now, talking about terbium-152 for PET imaging, so, and putting it in perspective with gallium-68, the half-life is pretty significantly longer. Then what's again is important is that it's got the same oxidation state where for chelation, normally we've already seen several chelators for gallium-68. For now, work has been done with DOTA for terbium-152. With regards to practical clinical application, with gallium-68 PET can be performed at really short times, as we already know, but again, with terbium-152, we can really perform late scans and actually perform predictive dosimetry in patients. Right, so this is about the production of terbium-152, and it's produced at Isolde CERN with spallation of tantalum with high energy protons following online mass separation, and goes into separation of terbium from its impurities, and eventually getting the final product, which can be achieved with a radiochemical purity of more than 98%. Right, with regards to radio labeling, if you compare the terbium-152, and then because the perspective of developing these radiopharmaceuticals or trying to work on them is the eventual concept of theranostics, so that we can actually apply this to lutetium-based therapy. So again, in these, here you can see that with these PSMA-expressing cell line, the uptake over time is pretty similar to lutetium. Now, this is your terbium PSMA 617 PET-CT. Again, PSMA positive cell lines on the right and negative cell lines, showing that this is pretty specific. Right, and here you can, there is an example animal work where you can see that there is uptake is seen at up to 2 hours post-injection, whereas relatively less, but you can still see specific tracer uptake on 15 hours images post-injection. And there's no significant renal uptake here as well. So with regards to biodistribution of terbium PSMA 617, again, you see that there is uptake, but as we'd seen in the images previously as well, it is not that high, whereas there is specific and comparatively higher uptake over 1 to 24 hours in the PSMA positive tumor cell lines, maximum being seen at about 4 hours post-injection, and then it goes down gradually up to 24 hours. With regards to tumor to kidney ratios, however, it over time, from 1 to 24 hours, it does go up, but again, it is similar to lutetium, and this helps in pre-therapeutic dosimetry and treatment planning. So the first case, first patient with terbium PSMA was performed at our center. Again, prostate adenocarcinoma. He received 215 megabecquerels, and image acquisitions were acquired anywhere from 50 minutes up to 25 hours post-injection. So these are the images of this patient over time, and as one can see, just enlarge this image where, I mean, because of the physical properties, as the current scanners are not actually, the software is not programmed to take up to actually comply 100% with the physics of terbium-152. Despite that, one can see tracer uptake in pathological lesions, which we actually compared to our previously done gallium-68 PSMA scan. Here you can see tracer uptake, which increases over time, even on very late images. So probably here maximum is probably at 18.5 and then later at 25 hours, but one can actually see this excellent uptake at such late time point. Similarly, in very tiny lymph nodes, here pelvic lymph nodes, one can see uptake as well, and up to the 25 hours. Another lymph node, and this is in comparison, which I wanted to show. So this is about 1.5 hours acquired, so late pelvic images on the gallium PSMA scan compared to the scan at 2 hours. And you can see almost equal uptake of tracer in both. Right, so this is again uptake in a lymph node here. The bulk is larger, but you can still see uptake on the terbium PSMA scan. So essentially, with regards to potential users, terbium is the only radiolanthanide suitable for PET imaging without the emission of beta or alpha particles. Due to its relatively long half-life, terbium may be of interest in combination targeting ligands of enhanced blood circulation. For example, when we are talking about thinking out of the box, and with the current success of theranostics, we're thinking about approaching newer entities for therapy. You could actually also attach them to antibodies, which is not possible when you're dealing with agents which are not that, which do not have a long enough half-life, and of course for delayed PET imaging. And as discussed a few times earlier, it may certainly be used for dosimetry prior to radionuclide therapy. The characteristics of terbium are essentially not ideal of 152, however, at this point, so one has to keep this into mind. And the production at this point is only available at the Asolda facility, and probably in the future they'll be expanding what else can be done with it. Do I have a few minutes more, or do I have a few minutes more this terbium?
Maybe one.
I'll try very shortly. So the last, and I'll try to rush this very quickly, terbium-161, it's of interest simply because we are actually going to perform some clinical studies following a grant from NetRF in collaboration with PI Sai Villigan. And this is interesting because terbium-161, along with beta emission, it also gives Auger electrons, so radiation. So this is very important from a therapeutic perspective. So shortly, we can understand that for the larger metastases that we see, we could make use of the beta particle. However, for the really, really small single cells, you could use the really short distance that the radiation travels to the extremely high LET in comparison to that. So not go into much details, but there are some images here at 1 hour and 24 hours of terbium, again with positive PSMA cell lines, and you can see in rotation some excellent uptake even at 24 hours post-injection. So I'll probably end that. There are some publications on that, and we could possibly just one slide on terbium-149, which could be used for PET-CT imaging. And then I'll thank our collaborators who've done the most of the work in the study. So thank you very much.
Thank you very much for this really interesting insight into new radioisotopes. There's a lot in development and exciting work. Time has progressed, so we will use a break for discussions. Thank you very much again. And please all return at 3:30, right, for the next session on liver-directed therapy. 3:30, please.